Good morning. I hope all of you were able to find your respective groups, and we'll be doing some clicker stuff a little bit later, and so you'll need to be sitting with your group as we go forward. We had gotten to the point of understanding a lot of these different components that are involved in regulating this one type of thing, which is transcription. And actually, it has to be a cooperative effort between epigenetic controls and transcriptional controls. So you can't get, you can't get this without the epigenetic controls cooperating with those transcription factors. Okay, so what we want to be able to do is we talk about different types of transcription factors, and we want to be able to construct a genetic switch to actually control transcription. And so that's what we're going to be doing today, is looking at how you put a combination of transcription factors together to do that. And then we'll look at how you can repress that transcription. There's several different ways. Okay, so we got to this big sort of disorganized, it almost looks like the San Francisco 49ers and the Ravens on top of them. It's a big dog pile. Okay, and so that's really what it is you have a unique, so this is gene X, there must be a unique combination of, number one, control elements, okay, and proteins that are bound to those control elements. Okay, so the control elements are basically the DNA. They're landing strips for different kinds of proteins, and this gene will not turn on unless the appropriate combination of proteins is there. Okay, and so you have all kinds of proteins assisting, but what you want to do is stabilize the general transcription factors that form the pre-initiation complex and hold on to pole 2. Because pole 2 is what you got to kickstart to get it to go down the template. Okay, so sometimes you use proteins upstream, way upstream, or very close, that actually bend the DNA. And if you want to bend DNA, you want to break our rule that is transcription factors bind to the what am I trying to say? The major groove. You want to bind to the minor groove, because binding to the minor groove puts a kink in the DNA and flips it over. And what that allows for is a region where these proteins can come together very closely and interact, either positively or negatively, and form what's called an enhanced cell. Okay, so that's what you want to do. And so to get to that point, though, you need to be able to open up the chromatin, because epigenetic controls are always dominant. They're either, if there's no open chromatin, you're not turning the gene on, because the transcription factors can't find their binding sites. Right? So this chromatin remodeling complex, if this is going to open up the DNA, a good example is one that's listed on your page. Is SWE/SNF. It's basically called a nucleosome plow that opens up the nucleosomes and reveals the landing pads for those transcription factors. So if this remodeling complex goes, and it can actually split ATP and actually move the chromosomes off of the chromatin or off of the DNA, revealing control elements. And then that's when these transcription factors can bind and start things going. Okay, you have histone chaperones that can actually remove the histone and transfer it to a chaperone that will grab onto it and keep it from unfolding, so that it can be quickly replaced. Because this is happening on millisecond timescales, and so you don't have a lot of time to do things. You take them on and you put them right back on. Take them off and put them on. Histone chaperone, you can actually use, you can insert isoforms of the histone, the core histones, like we talked about last time. Different isoforms of histones mean different things. The one that we talked about, which was SYNPA, is actually going to cause the reverse of this process, correct? Because it causes chromosomal condensation near the centromere. Well, there are other isoforms that actually open up the chromosome. What actually does that? Well, the histone chromosome chaperones are keeping it folded so it can be quickly shuttled in, but it's this remodeling complex that allows the DNA to unwind so that exchange can take place. All right, and then finally, you recruit histone modifying enzymes, that is, code writers. And they write the code that recruits the code readers that causes the thing to go forward. And so what we need to do is actually look at a very specific example of a gene that's being turned on and the sequence of things that happen. So this is the human interferon gene. Okay, it regulates a lot of immune cell function. Okay, it is a protein coding gene, and there's the Tata box, so that would be part of the promoter. And it has been unwound by one of these chromatin remodeling complexes so that this part of the DNA is now revealed. So we've got some of the epigenetic blocks out of the way, and we're going to recruit even more code writers so that we can recruit more proteins. So the first thing that actually binds is when this landing strip is revealed, it's either a promoter proximal element. It depends. See this little nick there? It's not telling us how close to the Tata box it is, because that would tell us whether it's a promoter proximal element or an enhancer. If it's way away or if it's way downstream, it's an enhancer, right? If it's within 1 to 200 base pairs, it's a promoter proximal element. Okay, so the transcription factor, they're not specifying what it is, but it sits down and it recruits a code writer. And that code writer is a histone acetyltransferase, and this one in particular is going to add that acetyl group, the H3K9 position. And remember how to read this. This is the H3 histone, and it's the 9th lysine. All right, and it doesn't do it just on one end; it does it on both of the H3 binding sites. How can it do that? If it's over here, how's it going to get over there? Any guesses?
And just sort of end around.
Yeah, is this thing a stiff Statue of Liberty? No, it's floating around. These things are waving all over the place, really fast. And so it's going to come in contact with that enzyme long enough so that you can acetylate both of those binding sites. Okay, so we're going to acetylate, and we've sort of had this feeling that acetylation is generally a transcriptional activator. So it's writing a code that will activate transcription. So the next thing that happens after we do that is the surface between the first transcription factor and the HAT enzyme, the histone acetyltransferase, forms a binding surface to which another code writer is going to bind, because we're not finished writing the code yet. Usually the code is not one simple mark. It's several combinations of marks. Okay? And so what this is a serine kinase. So it's going to phosphorylate H3S10 in 2 different positions. So now we've got a couple of acetylated histone residues. We've got a couple of phosphorylated histone residues. So we go to the next step. And what happens next is the HAT enzyme is going to phosphorylate a different position. So this is H3, still the same histone, but now we're going to phosphorylate lysine 14. Because it's the combination of these proteins has moved its binding site over where it can actually reach this particular position. And so it's putting another acetyl group. So now we have acetyl group, phosphate acetyl group on both ends of the H3 core histone, right? So that's what we needed. So that's the whole code that's written, and that's going to recruit 2 different proteins. The first one, and really importantly, is TF2D, which is the first thing that binds, locates to the promoter. So that was really key. It is a code reader. And the second code reader is a nucleosome plow, because you still got nucleosomes in front of you. Those have to be plowed out of the way, and that's what this thing is going to do. But it's a code reader because it's reading the code you just put onto that H3 histone. And so now you've got the machinery to go ahead and activate transcription. What else has to happen? All of the general transcription factors have got to assemble, and so you've got to have lots of interactions here before this can finally happen. So we're just following the very earliest phases of how you get started with this assembly. Okay? All right. And then here's a general model of if you're going to attack any gene. First of all, you're going to have chromatin remodeling complexes to open it up. Histone modification enzymes write the code. That recruits other activator proteins. Then you get So activator proteins would be TF2D and the SWE/SNF. Mediator binds, which is this big scaffolding protein that brings a lot of the general transcription factors all at once. And so they don't have to salt onto the dog pile one at a time. It's kind of like one of those fights we saw the other night. They all just jumped on together. That's what mediator does. It brings all those proteins together and sticks them on to the pre-initiation complex. Okay. And therefore you have transcription. Okay, so the reason we're beginning to know what all these different things do is because of this project. The Human Genome Project told us the actual sequence of nucleotides 3 billion base pairs long, right? We know the sequence. That doesn't tell us what all that stuff does, right? Remember I showed you that picture of a gene. Let's see, I'll show it to you here in a minute, but the take home from that message was the protein coding thing is really small, and there's lots of space with what we used to call junk DNA. What this project did was to try to understand what the junk DNA is, and it's definitely not junk. Okay. And so what they did was they mapped areas where transcription occurs, transcriptionally active areas are, transcription factor binding sites. So they looked at which of these control elements is recruiting a transcription factor, either positive or negative. So if it's positive, it's an activator. If it's negative, it's a repressor, right? They looked at changes in chromatin structure associated with that transcription, and they looked at histone modifications. They looked at how the code was written to mean different things. So they're doing functional studies. And what they came away with, and this is a huge consortium of many different labs, it's kind of the way science is being conducted now. It's big science. All over the United States and probably in Europe and elsewhere came together to attack this problem systematically, because this is too huge of a problem for a couple laboratories to take a stab at it. And so they've now been able to assign biochemical functions to 80% of that junk DNA. We don't always know what the exact function is, but we know, let's say, this region binds a transcription factor. This one is a chromosomal mark for transcriptional activation. So we're starting to put the pieces together, and this is a great first step. Okay, so instead of this kind of situation where we know what this stuff does, these are exons, and we know that this codes for a protein and that, but all this stuff here, that wasn't known. Now we know what most of this stuff does. We're 80% of it. So we're getting really close at figuring out a lot of new, and they've discovered all kinds of new novel DNA binding motifs beyond what I showed you during the lecture, but those are good ones to know, the ones I showed you. Okay, so there was somebody on Piazza that said, well, what are these codes? Do we have to memorize all these codes? And no, the answer is no, but let's look at a couple of patterns that you do need to know. So the next two slides, you do need to know what these marks mean. Okay? And the good news is it's not real complicated, because look at these first three marks I'm going to show you. They're all in the same histone, H3. They're all in the same lysine, K4. How do you read this? What's that mean? TV? No. It's one methyl group, right? So this is monomethylated, that's dimethylated, that's trimethylated. So let's see if you can discover a pattern by looking at this. So this is, if it's got one methyl group on that lysine, what it means is mark of regulatory elements associated with enhancers and other distal elements, but also enriched downstream from the transcription start. So if it's got one methyl group, what it's going to mean, you're going to put that mark pretty far away, but you're starting to mark your territory where transcription is going to occur. So it's distal. You see there's no promoters being marked, but there's enhancers upstream and downstream. So we're proscribing the boundary for what's going to happen next. Okay? Now, you got to identify the promoter, or you can't cause transcription, right? So you need to mark it this way. If you see an H3K2 dimethyl, that occurs mark of regulatory elements associated with promoters and enhancers. So it's also associated with enhancers, but more clearly, it's also pointing a finger at this promoter. So it's a histone, it's a nucleosome that's marked to point out where the promoter is. So you can see the more methyl groups we're putting on, to these lysines, the more closely we're pointing out where transcription starts. And then if you put 3, this is the pattern, okay? Mark of regulatory elements primarily associated with not just promoters, but active promoters, that is transcriptional start sites. So this is not a difficult pattern, is it? The more methyl groups that you put onto lysine 4, the more specific you're pointing out where transcription should start. All right, so that's one pattern that you should know. And what's the second pattern? Okay, this is just to remind you what's going on. This is a monomethylation, dimethylation. That's the only difference between these 3 marks, was how many methyl groups you put on that lysine. Okay, so let's go back. And so this is the pattern that we knew when this book came out, which was 5 years ago. A new edition is coming out very soon. And so these are all H3s. K4, well, they got it right? Certainly a methyl group at K4 does not mean silencing. So that breaks the old rule. We used to think that methyl groups were bad. Methyl groups silence, methyl groups repress. It's not true. So it depends which lysine residue you methylate and how many times you methylate it. If it's just 1, it's kind of far away from the start site. If it's 2, it's closer. And 3, it's right there on the promoter, and transcription's ready to start. Okay, so that still agrees with that little rule there. So that's really all we've learned, is that one position right there. And we don't need to learn a bunch to get the idea behind this concept. So here's a different one. Again, this is from the ENCODE project, H3K. So this is a lysine residue that's 5 residues away. And this time we're going to acetylate it, and that's a mark of active regulatory elements. Usually it has a preference for promoters. So what that means, we're going to find that nucleosome with that mark near a promoter. And the DNA should be open. It should not be in a 30 nanometer solenoid. It shouldn't be on a loop. It should be nice and open. And I'll show you what open means in here in a little video here in just a second. Okay, if K9, if you methylate it, particularly if you trimethylate it, that's a repression mark. So that's associated with constitutive, heterochromatin, and repetitive elements that have no business being transcribed. These are shut off. These are things that you don't. It's like it's not specifically the one we use for X chromosome inactivation, but it says don't use this. So this type of DNA would usually be found up against the nuclear lamina, where mostly you see heterochromatin. All right, so same residue, different mark. Thirdly, H3K27 acetyl group. This one's really interesting because not only does it mark an active regulatory element, it distinguishes which enhancers and promoters are active and which are not. So if you have this one there, not only is it the case that gets transcribed, it's going to be transcribed right now. It's a real current kind of mark. And so now we have 9 and 27. Let's see if that. Let's go back. So here, I guess if you methylate it, it probably does silence the X chromosome. But now we've seen that acetylating it is a really dramatic mark. That means not only is this a promoter for a transcribed gene, it's being transcribed. We're involving that actively. Okay? If this is methylated, can you acetylate it? No, you can't, right? You have to remove the methyl group. It has to be demethylated, and then you can acetylate it. Okay. The K9 position, see that was already on here, gene expression, so that was correct. A9, acetylated K9 is exactly what we expected. Okay, so we're now beginning to flesh out this really simple map all over the place. And so I've just given you a few examples of how to read the code. Combining all the elements of the code, that's a different problem. Okay, but it's being solved too. Any questions about what's going on here? So we now know that it's not simple enough to say, methylation is repressive, on histones, and acetylation is active, although we didn't find anything that contradicts the rule that acetylation usually means activation. That's still pretty much true as far as we're concerned. But methylation can mean a couple of different things. Now, can you methylate? Yes.
If we know the function for 80% of the DNA, why hasn't most of it been conserved?
Why hasn't most of it been conserved? What we do know is it's not the. We know the function meaning it binds a protein. We don't know what does it do. Mm-hmm. And certain of that proportion are highly conserved. They're under, we see them in primates and all kinds of animals all the way through evolution. Mm-hmm. And they're actually assessing that level of how conserved is it. But different animals do different things, right? Mm-hmm. So it's going to be different for us.
Right.
And so within primates, it ought to be conserved, but within, birds and reptiles, it's going to be less conserved.
Okay.
Yeah. I don't know. I'll have to think about your question more because that's a better question than I prepared for. Okay.
Well,
That's
I
okay.
imagine it's not one monolithic answer.
No, this thing is a book. It was actually published in September of this past year. And so people are still sifting through it. And just the list of authors is like 4 pages long. And yeah, so anyway. Okay, gene activator protein. Okay, so the other question I was going to bring up is, remember, before you can start doing all this marking around, you got to open up the chromatin. So, can you methylate DNA, and what does that mean? Okay. Generally, the rule is, so you methylate DNA at certain pairs of nucleotides, cytosine and guanine pairs. So cytosine, phosphate, guanine, that's a pair that can be methylated. And you methylate it at the 5 position on the cytosine, if you're asking. Okay, so it's the cytosine that gets methylated. Now, here's the rule. If you this is a really coarse, this is like a caveman's billy club. It just, it's really coarse. There's probably all kinds of cases where there are exceptions. But the general rule is if you methylate the DNA at their promoter, you're going to repress the gene. But if you methylate the DNA of cytosines within the gene, the coding region, RNA coding region, that's activation. So it is no longer the case that methylation of DNA is sort of a general blanket repressive scheme. We're finding even that rule gets broken. Okay, let's look at, here's a kind of a cool video from HHMI about that methylation process of DNA. And so let's see if we can do this. Let me turn this down. Oops, all right, so this is way too low. I'll start it over.
Here comes the protein. It binds.
No, it doesn't. Start over.
So here's a gene being transcribed, and next to it you see acetylated histone, nice open chromatin, and now you catch these little methyl cytosines. Methyl groups is appearing, and here comes the protein. It binds, and will attract this protein complex along with the enzymes, the deacetylases, and now acetyl groups are gone.
So there's the code writer.
Histones become tight, chromatin closes, no transcription factors can come through, and the gene is silent.
And what does that little piece remind you of? It's the start of a 30 nanometer solenoid, isn't it? And so you got to unwind that thing to get it to be transcribed, and that's just the reverse. And notice also, the methylation doesn't do anything by itself. What it does is it's a code that attracts proteins that recognize that signature. So again, let's up. So the concept then is the code is written on chromatin. That would be a fair statement. But you could say that the code is written not only on the nucleosomes, it's written on the DNA control elements themselves. Because chromatin, the word chromatin means DNA with associated histone and non-histone proteins. What's a good example of a non-histone that's associated with proteins? There's just dozens of examples I just showed you. All the transcription factors. If they're not a histone, they're probably a transcription factor of some sort, or co-activator, co-repressor, one of those dog piles. That's what the non-histones are. All right. Good. Where does it, let's take a, turn that, sorry. That's nice. Okay. Okay. Let's see. So let's ask a couple questions here. This was a question that I got on Piazza a couple years ago. See if you can figure this one out. There's no, you can't click it, but that's not what I meant to do here. And it's not a bad question. Okay, now get with your group and just come up with a joint answer to that question. And you can stand up, you can move around, you can whatever. I'm sorry we're so cramped, but
that would also be okay. Got an answer? Let's see. Let's take group number 8. Lucky 8 ball on the corner pocket.
We came up with the idea that the protein has both a DNA binding domain and a separate active site, a catalytic site. So it binds the coding strand and then uses a separate catalytic site to actively begin transcription on the non-coding strand.
Well, that's true. It's a big protein. It's kind of like that spaceship on Independence Day. It sits down way upstream, and it's actually way downstream as well. It's huge, all right? But does it actually bind the title box? What protein binds the title box? The title box binding protein. Okay, so this means, read that background if you didn't remember that. So it can't bind it directly because it's hidden up, it's hidden by the title box binding protein. But the other thing is it binding a strand? Remember our rule is that when the recognition helix goes in, what's it reading? Is it just reading one of those strands? What's the rule? I mean, it's sticking that recognition helix in there, in the major groove. It's making several contacts, right? It's not just reading one base, it's reading base pairs. It's reading both strands simultaneously, and probably the phosphate backbone as well, and the adjacent base pair next to it, and the one next to that. Okay, so it's reading double-stranded DNA before it unwinds it. And then it unwinds it, and like you said, the separate catalytic site, there's a cleft in there that the nucleotides have to come in, and it sorts which one matches this base that I've got underneath me, and then it puts the right one together, splits off splits ATP, it puts the monophosphorylated nucleotide right off into the right position, and then makes the next one, right? Okay, so probably the thing to take away from this is if you don't remember that kind of detail, then go take a look at that PowerPoint and outline for that background material. And this will be more straightforward. Okay, so moving on then, transcription is synergistic. What it means is a single transcription factor alone is probably not enough to get transcription going at any appreciable rate. So if you have a transcription factor that sits on one control element, you're going to get one small unit of transcription before that general transcription thing falls apart. Okay? If another one sits on, so this one is a different activator, and it's a little stronger, it's twice as strong, you get 2 units of transcription. But if you have 2 of them sit on both of their control elements, you get 100 units of transcription. Why do you think that's the case? Why are they synergizing? Remember what the goal is. Just think of the Super Bowl. The goal is to do what? Create an enhanced zone, right? And that's not 1, it's not 2, it's 3 or 4 or 5 or 6 or 7, 8, 9, 10, 12 different proteins that are all have to come together to stabilize the general transcription factor assembly into pre-initiation complex. So that's why they synergize, is they need help from one another. And so 2 is much stronger than either one of them by themselves. All right, so real simple concept, but it's fundamental. Okay, are we to repressor proteins? Yes. All right. So how do repressors work? We've already seen that you can write a repressive histone code, and you can write a repressive DNA methylation if you do it on the promoter. Okay, but this is different. These are transcription factors, and what that means, the word transcription factor means something very specific. It must bind to DNA. If it has no DNA binding domain, it is not a transcription factor. Can other kinds of proteins help? Absolutely, but that doesn't make them transcription factors. They're either co-activators or co-repressors. So both of these have DNA binding domains. And the way this repressor is working is that the sequence, the control element of DNA that it recognizes overlaps with the control element of this enhancer. And so they're competing for binding. And so it depends on who wins that battle. And who wins that battle is determined by the concentration of those transcription factors that are made in the cytoplasm and that make it back into the nucleus to compete. Whoever reaches there at the highest concentration is going to win that battle. Okay? So competing for a common binding site. So that repressor and the activator this is the silencer element, and this is the enhancer elements. They overlap. Okay, so you get competition. Okay, you can have them they're exclusive binding sites for each other. One's a repressor, one's an enhancer, but the activational domain. Remember, they all have this flexible neck connected to a protein-protein region that's going to interact with some other protein. If there's competition between these two, that means that the activator can't bind to its preferred binding partner to stabilize it. Instead, it's duking it out with this guy, right? And so they're having a fist fight, and so this guy can't do what he's supposed to do, which is to stabilize. Okay, so competition between the activational domains is a way of repressing transcription. Okay? Now, and they can compete for interactions with the general transcription factors. So if they're trying to bind to the surface of this TF2D, which is trying to get the pre-initiation complex assembled, and it's trying to get this is the promoter, and what's happening is this is going to prevent this guy from interacting with its binding site. So that the site of interaction may not be on this guy, it's actually, he's covering up the place that one wants to bind to. So it's the surface of all those pre-initiation complexes that it's covering it up. Right? So you can have competition between the DNA binding elements, you can have competition between the activation elements, and they can try to bind to the same surface and compete for that binding as well. So it's really all about competition, isn't it? Okay. And of course, you have repressive chromatin remodeling complexes that are recruited by, so this would be a good example of, so this would be a silencer, and then this is a repressor transcription factor that's recruiting the chromatin remodeling complex. And what's it doing? It's doing the opposite of the nucleosome plow. Instead of splitting ATP to loosen it, it's splitting ATP to wrap it up. So this is a very different, I think this is HP1, a remodeling complex that silences that gene. Okay. And another way to silence, so this is all at the epigenetic level. We're no longer at the genetic level, we're at the epigenetic level. If you're trying to fold chromosomes back into a tighter conformation, that's not a transcriptional, it's not a genetic control, it's an epigenetic control, because it has to do with the folding of the DNA, accessibility. And one way is to recruit a code writer that writes a repressive code. And one of the things that we know does that is a HDAC, histone deacetylase. Because what does it do? It's not writing anything. It's actually removing a positive code. So we still call it a code writer, but it's been recruited by this repressor to come here and write the code, make this, remove all those nice acetyl groups so that it can be wound together. All right, so code writers are recruited. Or, of course, this is just the same thing. This is a different type of repressive code writer. In this case, it's writing methyl groups on particular lysines. But be careful which lysine. You wouldn't put it on K4H3, would you? Because any of those three positions, 1, 2, or 3, on that lysine are all positive marks of transcription. It's going to put it on K9. Okay. Those are pretty similar. Any questions so far? So we have epigenetic repression, and so folding and unfolding, and we have genetic repression with transcription factor binding. And they cooperate. They're not independent.
Dr. Colley?
Yes.
Just genetic folding, would those be figures A, B, and C, and then from D, E, and F, would be then considered genetic transcriptional?
Well, I don't know what you mean by ABC, but...
Figures, A, B, C, so this would be...
Like that? See, that's a genetic control.
Okay.
Because it depends on the sequence of DNA.
Okay, so whenever you get down to the particular histone level, that's when the epigenetic.
When you're dealing with histones, folding and unfolding, then that's epigenetic. Or methylating the DNA, that's epigenetic. Okay. And actually, one can't occur without the other, right? You have to have a transcription factor, put a foot down and say, we want to repress this area. And so that's actually happening. So they're not independent by any means. Okay, and this is just another cartoon that I don't want to spend too much time on. But what's being synthesized in a particular cell determines what that cell makes. I mean, duh, right? And so here's the pool. And in this case, you have this protein and these three guys that these are transcription factors. These are activators. If it doesn't bind to DNA, but it's cooperating and activating transcription, it's called a coactivator. Not a transcription factor. It's a coactivator. It's not an activator because an activator is a transcription factor. Can you have co-repressors? Absolutely. Over here, the co-repressor is not binding to DNA, so we don't call it a repressor. These two guys are repressors, and that's the co-repressor because it doesn't. It's only protein that's going on. Okay. And notice something else, and this is crucial. This little green guy is a switch hitter, right? Over here, he's a he or she, whatever, sex. It's green. It's a green blob. The green blob here is a activator. Green blob over here is a repressor. Because this makes a unique surface that this thing recognizes. And so it's it's just a hammering into, it's a unique combination of surfaces that stabilizes the stuff at the promoter. Okay, that's the end of Quiz 1 material. So let's do a couple of things from Quiz 1, and let's take a break first. Let's do that. And don't run off because we'll do clickers and that's how I take attendance. Okay, let's get started again. So this is a question from last fall's quiz, and so let's take a look at this and try to answer it by yourself first. And you have to read that little tiny paragraph there, and then the answer is dependent on that knowledge of that paragraph. Okay, now get with your group. Well, no, actually, no, don't do that. Give me your answer now as an individual. And then we'll do the group session. There's no points. It's just, are you here today? That's really what I'm asking. But try to do it because you don't want to look dumb to your group. Bad idea. But there shouldn't be any coffee. Okay, this is individual.
Clicker's not going through, it's reading red.
Yeah, mine's not either.
Oh.
No, mine was working through.
I wonder what to do with this. Why doesn't like that? Why did I got 95 responses though?
Because E is the only one that won't work.
Yeah, 1, 3, 4 work.
Oh, Did I hit 4? Okay, let's try this again. We'll close it. And answer 3 is looking like a good answer, so in case you want it. Take advantage of that. 5. And just okay, try it again. And since you already should have the answer, then let's hurry up. Where's my clock? Ten seconds. Nope, there you go. There'll be another one. I'm not good at this. Okay, so most people, this is way too easy, I guess. So let's put that back up. Okay, so why is A wrong? So C is the right answer.
Doesn't each chromosome have its own?
Yes. And what's the DNA doing in its chromosomal territory usually? Is it awake or asleep? Sleeping. Okay. And so that makes this one wrong, but for this exact same reason. Even though it's homologous to that, they don't occupy the same territory. Homologous chromosomes are in different parts of the nucleus usually, which is kind of cool. This is true, so that's what you're looking for. And the activator B is a heterotetramer? No, not if it's a leucine zipper motif. Okay. Activator B dimerizes through its finger motif. No, it dimerizes through the leucine zipper motif. Yeah.
So if it activates or stimulates transcription of gene C on chromosome 2, would that mean they're close by but not in the same region?
No, they don't have to be anywhere close by. Remember, just think and loop out and go into a different part of the nucleoplasm and hit a transcriptionally active, what's it called? Hood. It goes for the hood, right? And so they don't have to be anywhere close to it. Okay, good. We learned several things for that. That this was too easy. All right, so let's go. Let's try this one. And do I have to tell? I think it should remember that it's 5. Okay, polling's open, and the only problem is you can't see that. Let me see if I can get that up there close enough.
You could delete 13 and not save it.
Good idea. So do it individually, and then. Give me some answer. Ooh, controversy. So, it looks like it's between B and D. Okay, so now get with your group and give me an answer. Let's see. How quickly get rid of this.
Well, remember,
So same question.
and so, control element A is on chromosome 13, and control element C is on chromosome 15. And so if it's binding chromosome 6, they're going to be different. Now, if I can I think it's because it doesn't make sense for the activator to find the probably going to do another transcription that will combine. So what is
Are active
Make sure everybody in the group that's when you're going to answer questions.
Why do you think remember what
Okay, we're waiting on a couple of
yeah, and so it's very likely.
Okay, 18. Come on, somebody's missing here. Okay, good. Who's answering? Let's, I know that's funny, but let's don't do that. Okay, because now we don't know what's going on here. So they're still equally divided. Boy, let's see. Man, we're going to have to talk about this one. Okay, that way. So was it B? It was B and D, I think, right? Or the two. So this one, control element A and control element both occupy the same transcriptionally act. So we're looking for the thing that's false, remember? And so there's a bunch of true things. So if it's true, we discard it. So that's true. You know, they've looped out. They're the same transcriptionally active. Control element A and C are located on different loop domains. Are they on different chromosomes? Yes, they must be on different loop domains. They can't be on the same piece of DNA. Okay, so B, it can't be the answer. Okay, both control element A and C are located on chromatin that has become less compacted. Yeah, that's right. Because that's how you have access to, you know, this is not simple or not too difficult. You just have to work through it. Activator B binds to control element A and to the major groove of gene C near its promoter. So we know it binds to control element A, right, because we're told that. And then it's going to bind to the major groove of gene C near its promoter. Can it do that?
No.
It would have to have, you know, two different DNA binding domains. And it can't do that. It can't bind this control element and activate that gene. So that D is the answer. And about half of you were correct. And answers B and C, no way. Okay. Good. Let's.
No, don't.
Oh, no, that's right. I've got a copy of that. All right. So, yeah. Now, let's look at it. So So this is the new material. So the Drosophila thing defines Quiz 2 material start place. Let's look at an actual example. And this is really, it's actually fairly simple because this is it's a silly fly, right? It can't be that complicated. But we've learned a lot from flies. So this is an embryo, it's a zygote, been fertilized a couple hours. There are 4 different transcription factors. There's bicoid and hunchback. These guys are activators. Giant and cruple, those are repressors. Okay, so that's what we know. And notice that there's different concentrations of these proteins in the embryo. So these are going to tell the fly where to put its stuff, where do the wings go, where does the head go, where does the tail go, right? And so what they do is they lay down a stripe of a single protein called Eve. Even skipped is the name. If a gene has a weird name, it must have been either Dr. Held or some other Drosophila person that named it. They're always jacking around with the names. Okay, so this is even skipped Eve, better known. All right, so you can see it. And what we're going to do is we're going to try to understand the control elements that will lay down that stripe right there. So we're going to have the right position and the right expression, the right gene expressed. So how can we possibly do this? All right, so it turns out that the information for where you put each of those stripes are all upstream from a single gene. That makes sense. You know, all the control over this gene is on the same piece of DNA, and so the module must contain the individual information for correctly laying down stripe 3, stripe 2, stripe 7. So it's kind of complicated to work with all those together. So what we do is we take a reporter gene that's easy to measure, that's not the Eve gene, and we're going to transfect this into a Drosophila, and we're only going to put one stripe 2 module upstream from that. So the rest of these are gone except for stripe 2. So we're looking at only one piece of DNA, and this is what happens. You get one stripe, and it's a blue stripe because that's what the LAC-Z gene stands for, right? It's no longer a stripe of Eve, it's a stripe of LAC-Z expression, okay? And so that matches this thing right there. And so how does it do it? So here's the structure of that module. Now notice a couple things. So the little railroad here, that's the DNA control elements, and with different colors, right? And can somebody close that? Yeah, thanks. And so the repressors are up top. They're just putting it there, and then they put the activators down below. So the little banana thing, that's a bicoid, and the little mushroom is hunchback, and the little Pope's hat, that's cruple. And then the Apple losing its value after Steve Jobs left. That's giant. Sorry. Okay. So all the repressors are up there and activators are down there. And so their binding sites are colored according to what they bind to. All right. So looking at that, you notice a couple things. There's lots of bananas. So there's lots of binding sites for bicoid, but there's only one mushroom. There's only one hunchback binding site. And you notice they're equally divided between the cruple and the giant places. So there's 3 of each, right? Okay. So, and you notice though that's the first thing to notice. And the second thing to notice is that some of the repressor elements overlap with the activator. So there's going to be competition, isn't there? At least there's going to be competition for both hunchback and giant and cruple are going to compete for bicoid binding. And here's a situation where the one control element to which hunchback binds is also a common silencer element. You don't need to say the word element. If I say silencer, it's an element to which giant binds. So there's competition between these. So that must mean that to get a stripe laid down, you need the activator to bind, and you need the absence of the repressor, correct? Because you see that if they're present, they're going to compete with the activators. So this is the winning combination. So this is where stripe 3 is laid down, and this is actually the position along the embryo, and this is the concentration of those proteins present in that nucleus. Okay, so giant this is its concentration profile, goes down, and you see there's no giant present where the Eve2 stripe forms. We know why now, right? Because if it's there, it's going to compete. We don't want it there. So we're going to lay stripe 2 down where there's no giant. And we're also going to lay stripe 2 down where there's no cruple. Right? There's no popat, right? So the popat and the half apple are gone. That's rule number 1. The second thing, notice, look, the concentrations of these two are very different. Hunchback has to be high, and bicoat needs to be low. Why is that the case? Hunchback high, bicoat low. What pattern do you see? Bicoat can be low. Hunchback, you have to have a lot of it. A lot of mushrooms.
The more hunchback there is, the less it competes with the giant, because it competes with the giant binding.
Giant's not there.
It's not
It would have to do that. You're exactly right if giant was there, but we've picked the stripe position where there's no giant.
It only has the one binding site.
Yes. Concentration is deciphered by the number of control elements present. Your book doesn't say that, but that's what the case is. So there's two things that have to happen. No repressors. No repressors. No giant. No cruple. Condition number, well, there's two conditions really. No repressors is one way to say it. And bicoat can be low. But hunchback has to be high because it has to, the hunchback concentration has to find one control element in all that mess in the nucleus. And there's lots of sites that banana, bicoat can find. So you don't need very much of it, right? Because it can hit several different places. So that's how you decode concentration distances. The control element number is important. Good. Okay. Any questions about that before we move on? Okay.
I'm sorry, I'm sort of confused.
Okay.
What does the Eve gene code for then?
What is the Eve gene code? It's a protein. I'm not sure. We'll ask Dr. Held if we can catch him in a nice mood. I really don't know what it codes for. It's probably. No, I don't want to say because I just don't know what it is. It's a pretty stripe. That's what it is. It's a brown stripe. Yeah. Okay. All right. So this is the takeaway message: is you got all these repressors and activators. The concentration is important. The number of control elements, where they're located, are they packed? So now you're building up quite a, story here of what has to happen when you want to turn a gene on and what has to happen when you turn a gene off, right? It's not that complicated. It's just, is it packed? Is it unpacked? How do you pack it? How do you unpack it? How many control elements? What are the concentrations of the transcription factors? So there's basic questions that you have to ask in order to figure out if that gene's going to turn on. Okay, and so taking up a more complicated situation, we don't need to learn this in detail, but this is the beta globin gene that makes hemoglobin, beta hemoglobin. And so we have 1, 2, 3 exons, mostly large introns. That's not important. There's the polyA side. This is the transcription start. The control elements are located upstream and downstream. One of the reasons why you got to put some of them downstream is it's kind of like the stripe modules. There's more than one stripe being laid down. In this case, there's more than one globin gene. Because as we go, the embryo expresses one type of globin and the fetus does another, and then early, at different times developmentally, you make different types of hemoglobin, right? Myoglobin is one of the early ones. Okay, so there's not a lot of space up here, so you got to put some of the control there and some of it downstream, and it looks just kind of like that other story. We don't need to memorize GATA and GCP1, whatever it is. A unique set of transcription factors has to bind and exclude any kind of repressor binding. Okay, that's really the rule here. Now, So here's that deal. The globin gene, globin gene is located there, and then these are other globin genes, various embryonic forms that are expressed, the gamma and epsilon, and there's no reason to memorize these. You can see they turn on at different times depending on which transcription factors are present in that cell, right? The really thing that I wanted to point out is this guy right here. This is a locus control region, and locus control regions are usually located 100 or 200 base pairs away. They can even be on different chromosomes. But their characteristic is that they loop out and they're bound to some sort of transcription factor that's required to activate a whole cassette of genes. It's going to activate epsilon, gamma, this type of gamma, delta, and beta. And so it regulates the expression of all of these hemoglobin genes. In fact, if you have a mutated locus control, the control elements of these guys are not enough. This person is going to have a form of anemia if that's mutated. Right? So the locus control is located a long way away, and it can activate each one of these. In fact, it must be there. It must be present. And it must be activated by some transcription factor. Because that's a DNA binding element, right? So there must be a protein that sits on it called an activator, and then it loops out and touches or associates with each one of these genes. Now, the interesting characteristic about the locus control region is not only does it have the ability to interact with the pre-initiation complexes of each of these genes, it also has a barrier protein. It keeps heterochromatin from encroaching on this region. Because you need hemoglobin throughout your life, correct? Or you die. And so you want to keep any kind of, say there's a spreading wave of heterochromatin folding into this area, locus control will put a bridge there and prevent it from being heterochromatized. So this will never become heterochromatin. So it's got two functions. It's got an activator function that's good for each one of these regions, and its characteristic is located way upstream of all of these genes. Second is it's unique because it has a barrier function. And we've already talked about the definition of a barrier. There's like 3 different mechanisms. Okay. Questions about what's important here? Again, our specific model is the Drosophila thing. Nice, straightforward, simple, but the mammalian system is not that different. Okay. And then we have insulator. So it's easy to get these two confused, insulators and barriers. With all this looping around, is it possible that you can have illicit contact? So here you have this locus control that's whipping out over here. What if it linked up with the wrong gene? It could. To prevent that, you have insulators, which are not the same thing as barriers. Okay, so look at this gene. Here's an enhancer. Here's gene B. This enhancer is supposed to turn gene B on. Okay? There's a barrier sequence that's preventing heterochromatin from encroaching in its territory, right? Now, the problem is this enhancer could, on part of the loop, it could whip out and it could interact and turn gene A on when you don't want it to turn on. So if you put an insulator sequence here, that prevents the enhancer from activating the wrong gene. How does it work? It's not real clear, but it seems to be that the insulator element actually is a decoy. It has binding sites that will bind to the enhancer and interact with it before it can touch gene A. Or it forms kind of a physical barrier. The insulator element will actually bind to the nuclear envelope, sometimes the nuclear pore, and form a physical barrier that the enhancer can't get past. That's kind of like what the barrier does, but remember, the barrier is preventing heterochromatin. This is separating active genes from touching one another inappropriately. Nothing worse than gene harassment. Okay, so about 2 minutes. So we've pretty much done this, and we did the invisible one, which is epigenetic control. And so we're going to say just a few words about RNA processing. You're supposed to know the details of capping, spliceosome, poly-A addition, know that they are put on there somewhere and where they're put. We're going to talk about alternative splicing when we come back on Thursday. Okay. So again, good thing, yes, did you have?
So, I have an announcement for you. Next week's gonna be the exam, right? On the
Right.
12th.
Tuesday, a week from today.
So instead of having my office hours on the day of the exam, I'll change the time and the place of my office hours. So in case you have questions before the exam, it'll be a quick contact.
And you're going to post those on Piazza?
I'll post it.
Okay.
There's nothing